This is a special episode of Bioactive because we are sitting down with the scientist who discovered the receptor in the brain that THC binds to, and therefore one of the most important people in cannabis science history. Her name is Dr. Allyn Howlett. I think a lot of people think that this happened a really, really long time ago, but it really wasn't that long ago in the grand scheme of things, because Dr. Howlett is still actively doing research and actively studying how this plant, these receptors, these enzymes, and these different molecules interact with the brain. And what you might not know about the discovery about how THC works in the brain is that a significant amount of this research into cannabis in the early years of research was completed by and funded by big pharma scientists. Also, many of you now know that I recently announced my pregnancy, so I will be doing an episode on my own personal journey, as well as talking to some medical professionals about cannabis and pregnancy on the show. I know this is a very stigmatized topic, but it's also a much needed conversation that I am more than willing to share at least my personal experience on, and of course listen to yours. I'll also be launching a few different research studies in the next few months to dive into this a little bit further. So stay tuned for that. And as always, make sure you leave a 5-star review wherever you listen to Bioactive, unless of course you hate women, weed, or science, but don't make me guess which one. So make sure you take a sip of water, stretch your legs, and just chill with Dr. Howlett and I for the next hour or so. All right, welcome back to the Bioactive Podcast. I'm your host, Dr. Riley Kirk, and I have a very special guest today, Dr. Allyn Howlett, who is a renowned neuroscientist who actually discovered the cannabinoid 1 receptor, or the CB1 receptor that THC binds to. So if you are somebody who likes to consume cannabis and you feel those effects in your brain, you can thank Dr. Allyn Howlett for being able to understand what's happening in the brain. This is an absolutely massive discovery in the neuroscience and cannabis space, and I'm so excited to have Dr. Howlett on to not only discuss the journey of discovery, but just talking about what that means and what has been done since that discovery in the cannabis space. So welcome to the show, Dr. Howlett, and thank you so much for coming on.
Thank you.
Now, would you mind just describing how you got into cannabis research in the beginning? Where did it all start?
Mm-hmm. Well, actually, when I started cannabis research, I had no interest at all in cannabis. I had just finished my postdoc and was very interested in studying a little bit about some of the neuronal hormones and neurotransmitters. And so my study area was using cultured neuronal cells. And at the time, the Nobel Prize winners for prostaglandins had just done some studies indicating that there were prostaglandins and there were receptors for those, prostaglandin D and prostacyclin. And I checked, and they were all prostaglandins that responded in my cells. So you may know prostaglandins are the compounds that are all involved in pain and other kinds of signalings. And when we use aspirin, acetaminophen, and other non-steroidal anti-inflammatory agents, what we're doing is blocking the synthesis of the prostaglandins. But there wasn't anything that could block the prostaglandin receptors themselves. So that was what I was studying. And in the process of doing that, I came across a paper from some chemists at Pfizer, Ross Johnson and Larry Melvin. They were involved in a big project at Pfizer Chemical to look for agents that could be used for pain relief that were not based on opioids, and they were not non-steroidal anti-inflammatories. And there's a good reason for that, because as we all know, the opioids can be very dangerous with respect to tolerance and physical dependence. And the nonsteroidal anti-inflammatory agents tend to be limited to pain that has to do with inflammation. So their goal had been to try to provide some kind of pain relief for people who wanted at-home pain relief rather than pain relief from a hospital setting. So they were very intrigued by the cannabinoids. There were papers that had been published in animal models, rats and mice, about pain relief. And so they were just one of 3 or 4 very large pharmaceutical companies who were pursuing cannabinoids. I think that most people don't even recognize that Pfizer and Abbott and we're all working with projects to utilize cannabinoid compounds for, in particular, for pain relief, but also for anti-anxiety and a couple of other things. So I contacted the Pfizer chemists when I saw their paper that said that they thought that because of the structural similarity of the prostaglandins versus delta-9 THC and its active metabolite with the hydroxyl group on it, that they were probably acting as blockers of the prostaglandin receptors. So,
because of the structural similarity, they said, okay, these probably act in a similar way, but maybe a different mechanism.
Just based on that. So I wrote a letter to them, and I told them that I had a model system. I was studying prostaglandin receptors, and I was very intrigued by their hypothesis, and I thought that I could test their hypothesis in my cultured cells. And I thought that was a very good idea because, if you put drugs into an animal model, you have to worry about uptake and distribution and does it get into the brain. But if you do cell culture, you're adding it directly to the neurons. So,
right, reducing those questions and just making sure you can figure out that little piece of the puzzle.
That's right. So they sent me not only the drug that I had asked for, which was the one they were talking about, but also...
So this is not THC. This is a different drug that they thought would be
similar to THC because what drug companies do is they take a compound, they try to make it so that it lasts a little longer in the body, is more active at a much lower concentration, that it's more specific for certain kinds of things. So they do a lot of manipulation to get their compound modified so that it's got better properties. So they sent me that. They said also that it gets acetylated for drug reasons. It lasts longer in a pill if it's acetylated. And so they gave me the deacetylated, which would be what happens after the liver takes the acetyl group off. So they sent me more than what I'd asked for. And I tested it, and it was not going to support their hypothesis. It didn't look like their compounds had anything to do with prostaglandin signaling in the brain or in the cell culture either. So I wrote them back, and they invited me to visit. Now, I should tell you that at the time, Pfizer had already dropped the project.
Ooh.
Because Pfizer's idea was that they wanted an at-home use because there were already a number of opioids that were used in the hospitals, but because of those properties of tolerance and physical dependence and sedation, and that they
just wasn't safe.
That they couldn't really put something on the market that would be that way. But they found that in their clinical trials, and they had done clinical trials on pain in hospital settings, that the better the pain reliever from all of the compounds that they made, the more side effects they were getting. So the side effects are the obvious ones. They are things like sedation or muddled thinking, And scientists call that cognitive dysfunction. And a few other things, maybe dizziness and loss of short-term memory. Now, these are things that really don't matter if you're in the hospital, because if you're in the hospital in a pain situation, you could probably use the rest, so sedation didn't matter. You're not doing anything important with your brain, but as an at-home analgesic, they felt that it wouldn't be a good market. So they officially dropped the project. Now, that was bad for mankind.
Yeah.
Because, if they had continued this, and this was, I believe, 1982 when they dropped the project, we wouldn't be having all of these problems now with should we legalize, should we not legalize, is cannabis a good pain reliever, is it not a good pain reliever? We would have had a legitimate pharmaceutical.
How crazy is that it just is really like hindering on that decision of a big pharmaceutical industry if this medicine works or not. Even though we have millions of people saying it does, it's still not accepted because way back then it was never done by a big pharmaceutical company.
Well,
that is insane.
That's right. Although the compounds that we have now include neuronal, which is delta-9 THC. It's just that it is made in its synthetic form. And we also have a compound by Lilly, which was made in that very same era. It's very similar to delta-9 THC, but it's got some modifications that make it a better pharmaceutical. And so it's been available for compassionate purposes all of this time, but it's now used in Canada, and it has been part of the pharmacopoeia here in America for some time. But, I think people don't really recognize this as medical marijuana, but these compounds were the first medical marijuana. On the other hand, in 1982, because the company did drop the project as a pharmaceutical, that made it very easy for the chemists to be able to offer those of us who do research a variety of compounds to be able to use in our basic science research.
Nice.
And they were very free about doing that because this was before the Bayh-Dole Act, which you may be familiar with, but that was an act of Congress that allowed universities to be able to make a profit on the research that was occurring in the universities. And once that happens, then you get concerns about patent protection and you get lawyers that are involved. But this was before that. So all I needed to do was write a letter to the chemist at Pfizer and say, please send me a few milligrams of the following items.
Of your finest chemicals for me to test on my
well,
assays.
Well, that's right. And, that was pretty standard procedure back in those days. Most of us got our prostaglandins from Upjohn, and they were, pharmaceutical companies were very happy to be able to give compounds to basic science researchers who could then provide the information that pharmaceutical companies needed to move forward. So that's how I got involved. And in the process of doing these searches for information, I found out, and I had been totally naive about this, I just assumed that since cannabis was in the 1980s a very highly used compound,
very.
that neuroscientists knew how it worked. But when I went to look through the research literature to find out what was available, there was very little information.
How cool. I love when there's just a huge open gap in research and you're like, oh my gosh, This is still so much opportunity here.
Well, there is, but I was not part of the a group of people who were really very interested in behaviors and the chemistry of cannabinoid compounds. So, I was coming in from the outside. And, I think that really does help. People come in from the outside without the prejudices and the biases of what they generally think.
And that can go both ways. Like, I'm certainly biased, but in the other way. Like, I'm so pro-cannabis, but there's also the other people that are so anti-cannabis that they can't really see straight and think of anything else other than drug addiction and reefer madness, you know?
Mm-hmm. So I was certainly helped by the chemists at Pfizer because they provided about 60 compounds. And that's how many they were making and testing. And so we could do what's called structure-activity relationships. So that means if you've got 60 different compounds that have small modifications in their structure, then you can decide what the best outcomes would be for what kind of modifications.
And you can compare and say, okay, this molecule has a little arm on it and it's acting this way in my assay. And this molecule has an oxygen on it and it's acting this way in my assay. And you can kind of start to piece together like what parts matter the most, which ones are most, doing the most, doing the least. I love SAR studies as a natural product chemist.
Yes. So I worked with the Pfizer chemists, and because the company was no longer interested, the value for research was that the chemists could get the data that Pfizer had on, they had 5 different assays, ways of testing pain in an animal model. So we could compare what I was seeing in my cell cultures with what they were seeing in these different assays.
Very cool.
Their data would never have been published if we hadn't pursued it together as a team, or rather 2 teams.
Right. And I mean, that is a nice collaboration of like big pharma and an academic institution, because you kind of have that method of checks and balances too.
Yes.
So you were still using the cell-based assay at this point for the prostaglandins, correct?
No, now we're using cell-based assays for cannabis.
Oh, I'm sorry.
Yes.
I'm sorry. The one that you developed for prostaglandins, but you're testing cannabis compounds on it.
Yes, but
okay.
We worked again with the Pfizer chemists. Now, the chemists had prepared something that could be made into a radioactive compound, because when the pharmaceutical companies need to send something to Food and Drug Administration, they need to provide the information on what happens when you give that drug in the body. So in that era, most of what we did was using radioactive compounds so that we could tell where they were in the body, where they are in the cell, and what they're broken down to after they've passed through the liver. So they already had a method of making something radioactive. Now they
were tagged. Sorry, they were tagged with a radioactive signal.
Yes.
So then if you either put it in your cell culture or you gave it to an animal, you could track how that was carrying
That's right.
through. Okay.
So if it was in an animal and it goes to the brain, then you'd be able to find it in the brain. Some compounds won't get through the blood-brain barrier, so that's an important thing to know. If it goes to the liver's got enzymes that will modify it. And we really need to know what those are. So, but the company had dropped the project, so there was no use for them for a radioactive compound. But there was for me. So they gave me the precursor, and I sent it to one of the leading radioactivity companies, which was called New England Nuclear at the time, and now it's part of DuPont. But they made a radioactive compound for me. And so we could use homogenates from rat brains, and we could use our cultured cells, and it would pick up this radioactive analog that was very similar to delta-9 THC called CP55940. 55940 is generally how I call it. So that compound now is radioactive, and anything that competes for it in its binding site, we can pretty much assume would be binding to that binding site. So THC was a very good competitor. But THC itself would not be a very good compound to radio-label for many reasons, because it wasn't nearly as potent as the compounds that Pfizer had made, number one. And number two, it's a very lipid-soluble compound, and so it gets into all the membranes of the cells and...
Yeah, we're just a big old lipid bubble, so
That's right.
It's really easy to lose lipids in a big lipid bubble.
That's right. But there were ways that we could modify the procedures so that we could identify this. So now we had a tag that we could identify a protein. And so that was really very useful for every researcher in the world. And so because DuPont made it, they could sell it. So
as they do.
Yeah, so it became available for worldwide use. So that's really the tool that it takes to say, this is working at a cannabinoid receptor, because if you're studying bone metabolism, for example, and you think that a cannabinoid agonist for the cannabinoid CB1 receptor is involved, then you can take your tissue and decide whether they actually have cannabinoid receptors, and whether that goes changes in pathologies or not. So
through the same method of, for example, with the rat brain, you would take that and homogenize it, mix it, put it in a blender, essentially, mix it all up, add your radio-labeled compound, and then you can see if that's essentially attracted to something or not.
That's right. So that's how you can tell that you have it. Now, some other researchers use that very same radioactive compound to do what we call autoradiography. And that means they could slice slices of the brain and incubate brain slices. And where the receptors showed up the brightest would, in the radioactive tags, that would be where you had the most receptors. And that's how we know that the cannabinoid receptors are virtually everywhere in the brain.
Were you shocked when you saw that, when you saw like it was all lighting up? Because I feel like because you didn't know about it before, you might think it was this like little niche part of the brain that maybe nobody knew about, but the whole brain lit up, Was that shocking to you?
Well, but some parts were a lot brighter than others. And those began to make sense because... and sometimes they didn't make sense. For example, at the time, using rat animal models, you wouldn't really think that a place like the cerebellum would have a lot of cannabinoid receptors because, for example, when alcohol is affecting an animal, you begin to see lack of balance, and humans, balance is something that the cerebellum does. But, on the other hand, the very early studies in cannabinoids used dogs as a model system. And they used dogs that had long legs. So they did have balance disruptions. So, that can account for that. There's a lot of cannabinoid receptor in the hippocampus, which is where memories are being laid down. So, those were very bright spots in the brain.
Yeah, there was a paper that came out years ago from veterinary medicine, and maybe it was what you're talking about too, that was, talking about that it's not safe to give your dog THC because your dog is not going to react the same way that we do because there's more of those CB1 receptors in the cerebellum that controls the movement and balance. And I believe it's called, what is it called? Is it ataxia.
Yeah.
When a dog like gets slightly immobile when they have THC. So that's just a little side note for listeners. Don't give your dog THC just because you like THC. It's different dosing. It's different products. You know, be safe.
Well, you know, we've come a long way, but we've used these same kinds of basic tools.
Wow, that is amazing. So this was in dog brains and rat brains that you were doing the slices with?
No, we just used rats and mice.
Okay.
And at the same time, a fairly large lab group under the direction of Dr. Billy Martin at Virginia Medical College at VCU, Virginia Commonwealth University, we're studying behaviors that were typical of cannabis use. And he developed a model to test 4 different aspects of animal behavior at the same time in the same animal. One of them is pain relief, as you can imagine. Another one is decrease in body temperature, because the cannabinoids always seemed to reduce body temperature by as much as 2 or 3 degrees, which is quite a bit.
That's a lot.
Also, a measure of ataxia, which would be more like balance, and then mobility in what you call an open field. So if you put the animal in a large box, a rat on cannabis isn't motivated to move very far. So we've got those 4 kinds of assays that can be done one right after another. And he used predominantly mice, but the same assays could be used in rats.
Oh, very cool.
Yeah.
Now, when you were getting this data, did you realize the impact of that discovery at the moment? Or did it take a little bit more analysis, a little more time? Or like when you published it and everyone was like, oh my gosh, this is world-changing evidence. Like this is massive for, medical cannabis patients, but also just, for the brain, for understanding our own body, a massive discovery. Did you realize that at the moment, or did it take a little bit of time?
I thought that it was very important to be able to demonstrate that there's a mechanism of action for the cannabinoid compounds, because at that point in time, the general idea was that they're very fat soluble, that they get into the brain cells and screw up the membrane fluidity and might act more like general anesthetics, which would be very nonspecific.
Right.
But this really demonstrated there was a specific receptor, specific signaling in specific parts of the brain, and that led to some legitimacy. It was no longer a mystery compound. It was something that researchers who were interested in virtually any aspect of neuroscience could now be able to develop good experimental design and study, the basal ganglia for motor dysfunction, or, endocrine studies in the hypothalamus, because now there was a reason, there was something that they could say it's a possibility that a receptor is now controlling this kind of behavior because it's working in this part of the brain. So that was very significant.
Did Pfizer come back and say, yo, wait a minute, we'd like to start this project back up because this is pretty cool data? Or what did Pfizer think once, they had ditched this project and then you found one of the most significant findings, in neuroscience history? What was Pfizer's reaction? Did they want to get back into cannabinoids and researching the ECS, or what was that reaction like?
I'll just tell you a little personal story.
Okay.
So I came back with my data on the 60 compounds to talk to the chemists about it again. And And they said to me, Pfizer really isn't very interested in this. It's, really just us.
Just for you, for your side project.
Yes, but at one of those trips back to Pfizer, I really couldn't really give a seminar or anything because it was the day that they were going to announce the launch of Viagra.
Oh, God.
So, that just goes to show you Viagra was so much more important than rethinking the idea of cannabis.
It's not surprising, though, but it is a funny little anecdotal story.
Uh-huh.
Oh, man. And so did you continue working with them at all, or was that kind of the end of the road with Pfizer? And then
well, the Pfizer chemists had been put on other projects. So,
okay, yeah, that makes sense.
But they had the foresight to say, I think since we can't work on this anymore, we really need to get together as a group of scientists and begin to coordinate activities. And so they helped find a chemist who was going to help from the NIH. That was Kenner Rice. And we coordinated with Billy Martin. So Pfizer gave them compounds for the animal behaviors at the same time that I got compounds to do the biochemistry and the cell culture kinds of studies. So we really did launch a more cohesive, larger group effort.
Oh, very cool. So after the discovery, you discovered this new receptor. It's the most abundant receptor in the brain, or GPCR in the brain, I should say. Obviously massive discovery, but there's a lot more to do too, right?
Mm-hmm.
So where did it go from there? And at what point did we discover endocannabinoids and that our own body is also producing these? And, were you involved in that research moving forward as well?
Not directly.
Okay.
The student that was working with me on this project was Bill Devane. And when Bill Devane got his PhD and left my lab, he went into Dr. Meshulam's lab.
So cool.
Yes, as a postdoc. And he was part of the team that was looking for what would be the endogenous compound. Now, they were not the only lab doing that. So,
I'm sure everyone was hunting.
Our lab had a project looking, but we were trying to assume, and here's where you come into problems. If you make an assumption and you stick with it and it's not the right one, then you're not going to make any progress. So the assumption in my lab was that it might be a peptide. And I think our lab wasn't the only one. There were other labs that were looking for endogenous ligands. And I think this was an era where we'd moved from small molecules like adrenaline and acetylcholine into the idea that there are a lot of small peptides that work as neurotransmitters or as neuromodulators. And so when you start purifying something, and you think it's a peptide, then the lipids, you eliminate those first.
Trash, yeah.
But the idea from Bill Devane in the Meshulam lab was, well, all these compounds that are cannabinoids are very lipid soluble, so must be the endogenous ligand must be a lipid soluble compound too. And while you might think that sounds, a little simplistic, they weren't throwing away the same parts of the homogenates that we were. So, we all got to the point where we were looking at some peaks on an HPLC, which is a high-performance liquid chromatography separation of things. And in my lab, we had a peak 10, but didn't have an identification on it. But in the Meshulam lab, they identified it. They had, obviously as a chemistry lab, they had a lot more equipment and tools to be able to identify small molecules. And so they identified the first one, which was anandamide. Now, Bill Devane was a very well-rounded person. He liked to read Hindu religious literature.
Oh, that's amazing.
So he came up with Ananda, which is Hindu for bliss, and amide because the compound they found had an amide linkage in it. So that's where the name Anandamide came from.
Oh, I'm so glad he named it. I'm so glad a cultured person named the molecule. That's beautiful because 2-AG is important too. I mean, maybe even more important, but way less sexy of a name. So people just don't talk about it as much.
Well, that's right. But, I do agree with you. It is more important because the anandamide, like THC, is not nearly as what you'd call efficacious. It doesn't work as well on signaling through the receptor as some of the compounds that come from drug companies like the Pfizer compounds. So they're called full agonists, and 2-AG is a full agonist. And it also is a lot easier for the cell to manipulate it immediately as a transmitter or a modulator. So,
to produce it on the spot.
Yeah.
It's easier for the cell to produce 2-AG compared to anandamide?
That's right. And it fits in with a lot of other cell signaling pathways that are in neurons. So it's like the final step in a signaling. But the advantage is that the 2-AG, when it's released, can come off the membrane and find its way to the membranes in the neighboring cells. So it can signal in the neighborhood of the cell that made it, whereas a number of other neuromodulators or neuropeptides have to be packaged into a vesicle.
Interesting. Yes, good point. And so at what point was the CB2 or the cannabinoid 2 receptor discovered after CB1?
Well, the interesting thing is the inklings of what could be a CB2 receptor started with the Miles Herkenham, who was the one that sliced the brains and did the autoradiography. And both his lab and my lab, so we did homogenates of tissues, and he did slices.
Gotcha.
But we both used the radioactive CP55940 to see what's there. And so we both got a lot of receptor in the brain, a little bit of receptor some other places. But he noted and wrote a paper on this, that in tissues that were immune tissues, you could also see some binding of tritiated CP55940, the radioactive compound.
Mm-hmm.
And we noted that too. We got tonsils from the hospital, and we were able to get binding in that kind of tissue.
Cool. Human tonsils from the hospital.
Oh, yes.
And maybe spleen samples too. I don't know if you're able to evaluate that.
Well, no, but you remember it was very popular to have your tonsils taken out when you were a kid back in...
Yeah. There's a lot of movies that have like weird scenes with people getting their tonsils removed.
And usually the kids, the best thing you could say to them is that you can have some ice cream after you get your tonsils taken out. So.
I can imagine if you told someone, hey, would you mind donating your tonsils to Cannabis Research after they're removed? Yeah, I guess I don't mind if my tonsils go to Cannabis Research.
Well, so I don't think we ever published that observation, but Miles Herkenham did publish his slices. So, so we knew that you could get receptor binding there. But, I think for a few years, everything was stymied with respect to other parts of the body because there is just so much cannabinoid receptor in the brain that if you try to compare it with virtually any other tissue in the body, it looks like a shadow. Is it there? Is it not there? Is it here today and gone tomorrow? So,
right.
But, much later on, one of the the aspects that I think moved the field forward was the discovery that there was an antagonist or a blocker. And that came from a small drug company in France called Sanofi. And they had done high-throughput screens with a vast library of thousands of compounds for binding using the radioactive ligand to lots of different kinds of receptors. So they weren't just looking at cannabinoid receptors. They were looking at radioactive compounds that bind to adrenergic receptors and dopaminergic receptors, the whole range of different kinds of neurotransmitters. And so they found what we now call SR141716, and that's an antagonist.
Did it have a drug name?
It did. It was
What was
Ramona Bont. And that was named after parts of the last names of the chemist and the biologist who had worked on it.
I didn't know that. That's so cool. I thought it was just a catchy pharmaceutical name, as we get sometimes.
No, the Ramona came from oh dear, I'm blacking on her name. The last syllable was from John Barth, who was the chemist. So
Okay.
so we have now something that can block behaviors in the body, different kinds of activities in other kinds of tissues. So that really moved the field forward, because now not only can you stimulate something, but if you don't know what it's doing, you can block it with a very specific blocker. So they also had one for the CB2 receptor, what we now know is the CB2 receptor. So now we could separate the two, because before that you couldn't separate CB1 from CB2 with the kinds of compounds we had, because they worked on both of them at the same time. So having the selective blockers could really tell that you had either a CB1 or a CB2 response.
Interesting. So Ramona Bont's kind of legendary because it was released as a drug, and there were some not in the United States, but in other countries, and there were some severe mental health side effects, so it had to be removed. When we were using Ramona Bont in laboratories, was that before it was released as a drug, or was that at the same time, or what was the timeline of those?
Well, I think when Sanofi discovered it, they reported it at one of our International Cannabinoid Research Society meetings, and of course everyone was excited. And so you got a lot of good research that came out from utilizing this compound. So I think from the company's point of view, they did what you would have done. You scratch your head and say, well, what can we do with an antagonist for this receptor? And so the question was, well, if people use cannabis and they get the munchies, and people who are obese have problems with eating behaviors, maybe we could use this to block the overeating that might be associated with weight gain.
Right. If it acts in the opposite way of THC,
Yes.
and THC gives you the munchies, you would think that an opposite THC would take away the munchies.
No,
but the
they
ECS is not that simple.
They moved very quickly into human clinical trials. So what they did was test people and they used a morbidly obese patient population. So they weren't looking at people who wanted to lose 5 pounds. They were looking at people who were in dire need of weight loss so that they wouldn't have cardiovascular problems. So in doing these studies, they noted that people did lose additional weight over and beyond what they might be losing with eating control, but mostly they noticed that the the change in their eating behavior was limited to the first few days. But the metabolic change and weight loss continued. So this was the first notion that we ever had that the cannabinoid receptors were involved in metabolism. So, going back and looking in a normal human or a rat model, on a normal diet, you don't see cannabinoid receptors in the liver. But in a high-fat diet in a rat, or in a patient who is overweight or obese, you do see cannabinoid receptors in the liver. So we know now that there's a mechanism by which high-fat diet can lead to induction of new cannabinoid receptors.
Was that the first piece of data that really made people say, wait, these CB1 receptors might be involved in a lot more than what we thought that they might be involved in?
Yes, I would say yes. Now, it could be that some other, like bone metabolism, those may have been going on ultimately at about the same time, but I think that this was just a major insight. It made a lot of people wonder, well, why didn't we ever see this before? And, sometimes things are right there in front of you and you just don't ask the right questions, and so you don't get the right answers. But it was the observations that Sanofi did in humans that really led to a lot of metabolism, not just limited to what happens in the liver, but what happens in the pancreas and the release of insulin and the fat cells. And so we know now that cannabis is the cannabinoid receptors are very important in storage of fat, which in most animal species, including humans, is a very important mechanism for survival. So these kinds of studies all came from that original observation.
Wow. I'm glad you shared that, because oftentimes when we talk about the story of Ramona Bont, it's really, really negative, and we're just like, that was a scary time. We shouldn't have given that to people, yikes.
Well, you know what? In the original clinical trials, and there were a large series of clinical trials. There were clinical trials for weight gain, clinical trials for blood lipids, clinical trials for cardiovascular aspects, hypertension, lots of clinical trials with thousands of people. But either initially one of the primary aspects to look for was weight gain. And in the people who are being treated for anxiety and depression, the drugs that are used to treat them always give weight gain. And so in their clinical trial restrictions, they did not want to have people who would be likely to be getting a weight gain, because that would defeat the purpose of what they were trying to measure. So they said that in the clinical trials, you could not be being treated for depression. So that's why it wasn't picked up in the thousands of people that went through at least 5 different kinds of clinical trials, is that if you had previously had depression and were no longer being treated, you could be part of that clinical trial. And so I think when they went back and looked at the data, there were some people who had problems with depression. But I think in the minds of the company, this was not a causal effect. It was more like a combination of events that could be an unfortunate combination. But, working on the, side of error, they didn't want to pursue this and put themselves in a situation where they'd have a liability. So the company took it off the market.
Wow, yeah, that just adds to my understanding so much more of that time. So appreciate you sharing that. So beyond the discovery of the CB1, CB2, of these different endocannabinoids, are there any other significant discoveries that you think really added to our understanding of the ECS? And or do you think there's still some big ones to come based on your research and other people's research? Because oftentimes in the community, we sometimes talk about an ominous CB3 receptor. Could that potentially be in our body somewhere? What are your thoughts on that?
Well, specifically with CB3, I think there are a number of receptors or other proteins that are modified by some compounds that come from cannabis. CBD is a floppy little molecule that can fit into a lot of grooves and places where it may not actually be the biological regulator, but it may have some, what you might say is allosteric sites. So it doesn't really bind to the receptor where its hormone or neurotransmitter binds to, but it modifies it a little bit. And it could be...
So not the main binding site, but somewhere else that affects it in some way.
And so the International Union of Pharmacology Committee really didn't think that meant that just because a compound from cannabis could find its way into a groove, that doesn't qualify it for being a cannabinoid receptor. Now, my preference is that there are a lot of compounds now that we know about that have that amide linkage, lipids with something else, some small molecule or amino acid or something. And there are a lot of compounds that look like 2AG because, you've got what I'm going to call lipid mediators. But just because 2AG and anandamide are lipids and their functions are clearly related to the binding site that is the main regulator of CB1 and CB2, these others are not really necessarily working the same way. They're modulating in a slightly different way because of the nature of the floppy CBD, or maybe the THC itself can fit into some place and modulate it. So that's why the IUPAR Nomenclature Committee has been very reticent, because some of these compounds, like GPR55, have another kind of lipid that binds to that its main site. And so, we can't steal the fire of the people who study those kinds of lipids and say, but that really should be called cannabinoid, because sometimes
it's ours now.
some of our analogs can bind to that site as well. So I think that CB1 and CB2 are what we call highly conserved. That means that no matter what species you're looking at, that protein hasn't changed from species to species. It is very important in most of the cells of the body as a feedback mechanism, whether it's a neuron feeding back to another neuron, or whether it's a storage property in the fat cells. These are all very important functions. And so as bodies have changed and species have developed, that receptor has maintained itself. And I think that's why we only have CB1 and CB2.
I think that's a very, very good answer. And I think that you just highlighted something so important, that all animals, including us, have these CB1, CB2 receptors because they're essential for our life and for our survival and for our body to talk to itself. It's a retrograde signaling system, and it's the only one in the body that can speak backwards and actually regulate things that if they're, overstimulated or understimulated, the ECS can kick on and signal to fix that. So have you are there any animals, invertebrates or vertebrates, that don't have an ECS?
Interestingly, insects.
That is interesting. Do they have some sort of parallel version of an ECS, or do we know any way of
I think
how
they handle lipids very differently than we do. And
so no insects have an ECS?
They don't have cannabinoid receptors, and they don't make anandamide and 2-AG. So I guess my answer is no.
Yeah, no, that's really interesting, especially because, some fungi, like the truffle mushroom, is producing anandamide or 2-AG, these endocannabinoids, and that likely might be a signaling to maybe the predators, I guess I would say, the foragers that are looking for them. I don't know what the role is in a fungi, but I find that really, really interesting. But I didn't even know that insects didn't have an ECS. Makes me feel, a little better about if I have to kill a mosquito or a fly or something, Awesome. Well, we're getting close to the end of this, and I just wanted to ask you, what research are you doing now? Because are you still involved in any research? Are you still consulting on people's research? Are you still teaching? What is your role right now with cannabis and the ECS and kind of passing on that knowledge and all of this, amazing information to the next generation of researchers?
Well, I think that since we only have 2 receptors and they seem to be dispersed throughout the entire body, we have very little way of selectivity. But, we do know that if you do use THC, you get different effects in different systems. And so the question is, why would an effect that might be in the liver or be different from what might be going on in the brain? Or is there a way that we can separate effects that might be going on in the neurons of the brain that regulate pain versus those that regulate memory, for example? So I think the answer to that, in terms of the cannabinoid receptor regulation, has to do with what kinds of proteins the receptor itself is interacting with. So we've been working with a cannabinoid receptor interacting protein that it's not the same as the G proteins, and it's not the same as the beta-arrestins, but it seems to regulate how the cannabinoid receptor signals. And because it's not made universally in all the cells that make cannabinoid receptors. So it can probably only function in the kinds of cells that it's made in. So since it's not made in all the neurons in the brain, we might be able to use that as a way of targeting a different kind of a target, not the cannabinoid receptor itself, but the associated protein to regulate something that might be beneficially regulated by a cannabis agonist.
So you could have very targeted medicine to a very specific part of the body instead of, taking an edible and feeling it all throughout your entire body. That would be amazing, especially for people who have chronic pain but don't want to feel high all the time.
Yes.
They don't want it in their brain, but they do want it in their body.
So what we need to know a little bit more about is where are the associated proteins made? And this one that we're looking at is not the only one. There are several associated proteins that can modify functions in different kinds of cells. So that gives us a little bit more insight into at least the basic mechanisms of why it might work one way in the brain, but a different way in another kind of cell, or maybe one neuron versus another kind of neuron.
Oh, that's fascinating. I'm excited to stay up to date with this research and learn more as you continue to publish and your colleagues continue to publish. I want to thank you so much for taking the time to come on the show and educate me, educate the audience, and tell your story. It's really fascinating. And the fact that this didn't happen that long ago, this is relatively recent history that really changed our view of neuroscience and cannabis. And I just feel so honored to have the opportunity to talk to you. So thank you so much for coming on.
Well, thank you, Riley, for having me.
Oh, absolutely. And if you want to just share, if you have a lab website, an email, if you want to share, you don't have to, or any other way people can keep up to date with you, that would be great. And then we'll end the show.
Yes. Well, I work with the International Cannabinoid Research Society. So I would say that people who are really interested in cannabis research it would be beneficial to them to pay attention to what's going on with all of the researchers at the International Cannabinoid Research Society.
Okay, ICRS is the absolute elite way to stay up to date with cannabis research. And they still have yearly meetings, right? It's not every other year? It's yearly. Okay. It's every other year. And I think it's international. I forget what country it's in this year.
Bloomington, Indiana.
Oh, it's in the U.S. this year. Yes. Very, very cool. And sometimes they offer virtual passes as well. I'm not sure if they're doing that this year, but definitely stay up to date there. And thank you all for listening. And thank you, Dr. Howlett, again for coming on. And we'll catch up with everyone in a couple of weeks. So mad love.
Thank you.